what you grow, the source of growth, the medium that's growth used for growing these things, they can divide every 20 minutes, which is very rapid for microorganisms. They are, and I'll give this term now because we'll talk about it. And what I should point out to you is that when I do these microbes of the day, I'm going to be introducing concepts of things that might not be necessarily introduced for a couple of lectures. So when I do this now, it means that I'm focusing on it, and I want you all to know it, even though I won't be talking about it in lecture for a while. This one term, though, for E. coli is actually something we will talk about. That's not where I want to go. So where's my mark? Where's my pen? So this organism is a facultative anaerobe, and it is also a chemoorganotroph. Okay, so chemoorganotroph, you guys should know. Does somebody want to define that for me, since that was part of our reading, the reading of this chapter? So thank you, Sravi. Yes, they get energy from degrading organic compounds. So the organo part refers to organic compounds, and their chemical energy comes from degrading organic compounds. Also, they derive their carbon from organic compounds. That's also really important. An organism that is a lithotroph, lithotroph, is one that's going to be gaining its carbon and energy from inorganic compounds and not organic compounds. Okay. E. coli, on the other hand, is, well, actually, no, they rely on Ravdeep, they not necessarily rely on the metabolic activities of photosynthetic organisms. It's going to depend on, that's a direct or an indirect statement. We'll talk a little bit about that in a bit. In general, yes, but there are organisms that do not rely on photosynthetic organisms whatsoever to get their carbon, their energy. And those are organisms, for instance, that live in dark environments. They get their electrons, their energy from inorganic compounds. So let me get rid of this because that's not E. coli. Facultative anaerobe is an organism that grows in the presence of oxygen, but will also grow in the absence of oxygen. So facultative means that it has the ability to grow both aerobically and anaerobically. The anaerobic means that when it says facultative anaerobe, means it rather grow aerobically, but it can grow anaerobically. So that's E. coli. All right, so the pictures of the plates here of E. coli growing on it. So if you're not familiar with this, this is basically media where the E. coli has been spread on it, and the little bumps here, each one is a colony that's derived from a single cell. And I want to point out here that this is pink, and you have one here that's half is pink and half is yellow. So let me talk about this for a few minutes, because we'll talk about this a little bit more, but I want to introduce it here because this is a way of distinguishing E. coli from other bacteria and other types of E. coli that are similar. Sorry, other types of bacteria that are similar to E. coli. So let me get a screen here to write things. Okay, so this media is called, I'm having a lot of trouble with it. There we go. Okay, this media is called MacConkey agar. This is a very, very specific type of agar. It's the most common agar that you find in clinical microbiology labs because it's got both properties that make it differential and selective. Okay, so it has the ability to differentiate between different types of bacteria and has the ability to select the growth of different types of bacteria. So what that means is that when we look at the composition of this medium, this contains protein. So it has a source of protein. It has a source of lactose, which is a sugar. So this is a carbohydrate source. It has in it bile, so something that's produced by our livers for digestion. It has a crystal violet, so the dye crystal violet. And it also has in it a pH indicator called neutral red. Okay, so all of these components are what are used to provide the characteristic medium as differential or selective. So only gram-negative organisms can grow on MacConkey agar. Gram-positive cannot grow on them. So it differentiates, it's selective for gram-negative bacteria. Gram-positives will not grow, and that is because gram-positives are inhibited by the bile and crystal violet. So the bile and crystal violet inhibits growth of gram-positive organisms that have to do with the peptidoglycan synthesis, etc. So that's why this medium is selective for gram-negative bacteria. So that's the selective part. The differential part has to do with the difference in the source that the organism uses for growth. So bacteria that can use lactose as a carbon source will metabolize it, they'll eat it, and as a consequence, they'll produce acids. The acid production lowers the pH, and because you got a pH indicator in there, the medium turns red or pink. Bacteria that cannot use lactose use as carbon source will use the protein. And protein is not a great carbon source. It's actually pretty poor, which is why the bacteria that use the lactose will use that first. So that's why that's a preferred source for metabolizing and getting a carbon. Remember, protein is made of amino acids, and amino acids have carbon in it, and so you get the carbon out of that. What that does is that results in the production of ammonia from utilizing the amino acids. Ammonia is basic, increases, so this is basic production, base production. That increases pH, and medium turns yellow. Okay, so you can differentiate between the organisms that are using lactose, the ones that can't with this medium, and you can select for gram-negatives over gram-positives. E. coli, and I'll leave this up for a minute and then go back to the slide. So E. coli is both a gram-negative bacterium and uses lactose as a carbon source, so it's going to make the medium pink or red, and that's how we know that it's E. coli there, among other characteristics. There was a question: is there a way to see typing sooner while you're typing? Well, you're seeing the typing as I'm typing it, so I'm going to leave this here, and you guys can write this while it's actually going to be on the recording too if you miss it. Okay, does everybody see this? Okay. Great. So any questions about the difference between differential selective and what McConkey algorithm is used for? If you were in taking 273 lab right now, this is one of the ways you would be looking. I believe it used to be this way. I'm not sure what they do anymore, but McConkey is a very, very straightforward way of differentiating gram-negative enterobacteria from gram-positive and other enterobacteria. So in general, differential and selective media, yeah, this is specific for McConkey, right? General differential medium is one that can differentiate between different types of bacteria, and I'll be showing you these in a couple weeks. Selective are also very specific media that can give you growth of some bacteria over others. And if you go through, I'm not sure you can see me through the camera, but there's a book called the Diffco Manual. It's made by a company called Diffco, and it's got about 1,000 different types of differential and selective media for different types of bacteria. Okay, does everybody have any questions about this? So I can move on for 2 more minutes. Okay, so let me go back to the slides. And just a couple more things I want to say about E. coli. So in general, E. coli is a beneficial organism. It's present in the gut. It provides us with some vitamins. It also provides us with the ability to colonize the gut so that it keeps it away from other pathogenic bacteria, so it's a beneficial bacterium. There are some strains of E. coli that are not beneficial, and there are some that produce toxins. So there are some that are exotoxin. They produce what are called Shiga toxins. These are exotoxins. So these are released from the bacterium, and releasing them will result in gastrointestinal problems. You end up getting bloody stools and things like that. These are things that you get food poisoning from food that's not been prepared, and you have a very specific variety of E. coli. The ones that usually appear from, for instance, eating at McDonald's or eating at Wendy's or something, where they have tainted salad or tainted beef, are strain of E. coli that is called and let me write that down. It's called the H. You might have heard this before. Sorry, the O157H7 strain. This is a particular nasty strain of E. coli. The O stands for a type of polysaccharide that's being produced on the outside membrane, and so part of the lipopolysaccharide. And the H refers to a particular protein in the flagellum that is characterized of this bacterium. This is both an endotoxin and an exotoxin. The exotoxin is the sugar that's made. The endotoxin is this structure here that cause diarrhea and causes all kinds of gastrointestinal problems. Septicemia, apparently, sometimes, and urinary tract infections, etc. So serious poisoning. But in any case, most Escherichia coli that you find in the environment and you find in the gastrointestinal tract are beneficial organisms. Okay, any questions about this microbe of the day? And it's spent more time than I want to, but I wanted to just focus on the McConkey. Again, here's the example on the right side of this is the organism that's plated on the right here is not E. coli. The one on the left is, because that's pink. The one on the right is a different bacterium that can't use the lactose in the McConkey. Okay. Any questions about that? Okay. So let's go to the next slide. I have a poll here that I'd like to do. I want to do these occasionally. Let me see if I can get this. So here's the slide. Motile cell having no visible nucleus was isolated, yielded purple cocci with gram stain. So you can assume that the cell is one of these, A through E. And I think I haven't done this before. Let me try to see if I get the poll going here. Hello. All right. I'm afraid that it's probably covering the answers here for you guys. Is that true? But you can go ahead and start answering. I hope you can do that. I'm sorry. So let me repeat D and E. So D has a cell wall, and E is capable of producing endospores. So you're saying I can close the slide and open it again? Is that what you're telling me?
Professor?
Yeah.
The students can press the X on our box and close out the poll, and then we can also reopen it and answer.
Oh, okay. Thank you. So I don't have to do anything. That's wonderful. All right. So I'm going to give you 1 more minute. And let me end the poll right. 16 of you need to answer still. Just if you don't know, just guess. I want everybody the opportunity to answer. Okay. So, if you answered is gram-negative, you're correct. If you said it has a mitochondrium, why would it not have a mitochondrium? So it can't have a mitochondria, right? Why should it not have a mitochondria? Right, so it's prokaryotic because it doesn't have a nucleus, so it doesn't have a mitochondria, exactly. Does it not, would it produce flagella or not? And the answer is basically in the question here. So, does it possess flagella? Yes, it's motile, so it has to have flagella. It has a cell wall. So I misspoke when I said this. It's a purple. I was reading this and saying something else. So it's a purple cocci. So it's not gram-negative. It's gram-positive, right? So this first one is wrong. The second one is wrong. It's not possessed with flagella is wrong because it's motile. It has a cell wall because all bacteria have a cell wall. We're talking about peptidoglycan, right? This last one here is capable of producing endospores. So endospores, as I said, are usually a hallmark of gram-positive organisms. However, I also said that gram-positives, not all gram-positives produce endospores. And so you can't assume that it's producing endospores for this last one. So the answer is D. And if you answered D, that was the correct answer. Okay, anybody want me to do that again? Well, if it had endospores, actually, you don't need to do an endospore stain usually to see endospores. If you use a light microscope that has refractability, the ability to do refraction, you could actually see endospores because they refract light differently than the cells do. There's a lot of ways of looking at endospores, but yeah, that you can stain them as well. D is not wrong, D is correct. So it has a cell wall. The only one that's correct here is D. All bacteria, both gram-negative and gram-positive, have great cell walls. Gram-positives are thick, there's thick peptidoglycan. Gram-negative are thin between the outer membrane and inner membrane. Okay? All right. The learning objectives for learning outcomes for this chapter are here. I'm not going to spend any time on this. Obviously, you can go into your book and you can look at these and write them down and answer them or study them in this way. They're available, obviously, in the presentation, so you have them for you to work with. Your book has things mixed in a lot of different places. I'm going to do it in a little different order the way they have it, although it's pretty much the same. And so hopefully by the end, everything will make sense to you in terms of how this works. As I mentioned to someone earlier, this is a lot of material, and it's important material for microbiology, well, for a lot of things. So we've separated this out into 3 lectures, and hopefully we'll cover everything we need to make it all clear for you. If at the end of the 3 lectures you still have some problems with this and it's still fuzzy, please contact me. Please make an appointment and we can talk about it. Okay. So I have an empty slide here. This is probably telling me something, but not sure what. Okay, so we're going to spend time talking about fueling reactions, metabolism, and energetics for today. Mostly the 2 lectures for next week, Tuesday and Thursday. So metabolism, your book defines this as sum total of all reactions in the cell, both the energy harvesting and the biosynthesis reactions. So the question is why, I mean, we're doing this. I said it's really important. Why is it important to study microbial metabolism? Anybody have an idea? And you can raise your hands, use your mics. I prefer that way instead of putting everything in the chat, but you can do it in the chat too if you want to. Okay, so Mackenzie said can halt it for medicine. I think what you actually meant is that you can use strategies if you understand the strategies of metabolism of pathogens, you can use that for ways to stop the growth of pathogens. I think that might be what you were saying. Yeah. Okay. Anything else? So that would be the medical reason, correct? Although there's another reason too. Helps to understand the pathways of energy in the body. Well, that's how you would understand metabolism, human anatomy, human metabolism. I'm actually more interested in microbial metabolism. Nutrient cycling in the environment is actually good. Certainly, you can manipulate the environment by understanding how organisms interact with their environment by nutrient cycling. That's right. Byproducts of metabolism are useful in other forms of life. That's absolutely correct. So you manipulate them. You are basically allowing them to produce beneficial processes like antibiotics, like wine, cheese, beer, food processes. Anything else? There's one actually really main one that's really important for my understanding microbial metabolism, and I actually sort of hit on it when we talked about E. coli earlier. And think about the differential characteristic of McConkey agar. So just to move this along, so it's actually a really good way of aiding in identification. So all bacteria have their own metabolic pathways, their own types of sources that they use, things that they make, things that they can't make, and you can identify organisms that way. The other important thing is that the human body has 10 trillion cells approximately, and there are anywhere from 10 trillion to 100 trillion bacteria on the human body, meaning that our body could be made up of as much as 10 times, the cells in our bodies could be much 10 times bacteria rather than our own cells. It's estimated there's somewhere over 500 to 1,000 species, and they weigh about 2.5 pounds, 2 to 5 pounds. Those are big estimates. So the fact is, though, since you're living with these things, you want to know something about how they eat, how they live, how they grow, how they survive. On the planet, there is in the range of 5 million trillion bacteria on Earth. So it's 5 times 10 to the 30th, or 5 no million. So 1/2 of the life on Earth is bacterial, which also means that you really want to know how these things are working if you're sharing your space with these microorganisms. So requirements for growth. So your book has this nice little picture showing the uptake of energy from the sun. What is that energy called? So that light energy, is it kinetic or is it potential energy? So the energy that's harvested by photosynthetic organisms, this is kinetic energy, right? Potential energy is what gets stored in the chemical compounds that these things are produce from that light energy. So kinetic energy basically is thermal, radiant, sound, electrical, energy of movement. Potential is stored energy. And I'll be happy to write this. Kinetic So basically, the radiant energy that's produced by the sun and produced by a lot of other things, not a lot, but other things, and we'll talk a little bit about that, is stored in compounds that are produced by photosynthetic organisms. Okay, so I showed wheat here, but that's because it's a crop that gets eaten. And then those things are eaten, and the energy that's stored in the organic compounds are then taken by organotrophs, and they're utilized to synthesize building blocks, and then produced to make macromolecules for growth and replication. So so that's really all I wanted to say. That so basically, what we have here is a series of events that occur from obtaining that energy source and breaking it down. So we'll talk about glucose, but there are other energy sources that the microorganisms use. And through catabolism or breakdown, of those energy sources, the potential energy that's stored in them is then released. And the release of energy, anybody remember what that term is called? Right, exergonic. The exergonic reactions release energy, and the opposite of that require energy for the synthesis are called endergonic, correct. So this really annoys me because I gotta select this thing each time. Okay, I'll get used to it. Oh, lost, ran out of room. Okay, so endergonic reactions are those that require energy. So let's just focus on the exergonic or the catabolic reactions. Oops, sorry about that. Catabolic reactions. So in effect, for the catabolism, and this is true for all living organisms, you're going to start with some energy source. The energy is going to be derived from it in a variety of steps, and those steps will result in gaining energy, or taking that energy the potential energy that's in the bonds of that compound and using that, as well as using that energy, as well as upon catabolizing that compound, you're going to end up with precursor molecules. Those are molecules that are going to be needed for downstream processes. As a consequence, there's going to be things that are going to be produced. So there's going to be waste products and CO2. When we release CO2 by exhaling, we're releasing the waste products of a catabolism. On the other side of that coin, you have the anabolism, or biosynthesis. So that's another synonym for anabolism. And the energy that's obtained from the catabolism is then used to produce macromolecules. It's used to produce all kinds of precursors that are then taken all together to give cell structures that allow a cell to grow and divide. And again, remembering that in order to do that, you need energy. So some of the energy used that obtained from metabolism has gone into biosynthesis, and then some of the energy that is obtained from catabolism is also used for a lot of other processes. Like, anybody want to speculate what other processes you need? A microorganism could use that energy for them if it's not going to be using for biosynthesis. Movement, yes, that's right. So movement, bringing things in, transport. Remember we talked about transport processes that are require energy. So that's where some of this energy occurs, is used for. Okay, and the routes by which these the process occurs via a series of different kinds of reactions. And this is rather straightforward, and you've seen this in the book. This is basically metabolic pathways that can be linear, they can be branched. So the intermediates that are made, and again, starting compounds are also known as precursor metabolites. And the intermediates here could also be referred to as precursor metabolites to make end products. And then you can also have cyclic pathways, and we're going to talk about a couple of cyclic pathways in the next week or so. And for your entertainment, or your enjoyment, or just to get a feel for this, you can go to this link, and you can pull up this chart, and this is basically all the metabolic pathways that are known in most of the organisms that have been described. There's some missing here that are found in the archaea, but in most cases, these are metabolic pathways. And what's really cool about this is they all basically center around one major cycle. So you can see right in the middle here, there's this big cycle here. And if you've had biochemistry, actually, if you go to that map, if you go to this link, you could actually zoom in on different places. And the places that you zoom in on are going to be, it's going to be expanded and see that better. Anybody have an idea what this cycle is here? If you've had biochemistry, or if yeah, that's correct. It's a TCA or Krebs cycle. And if you don't know, it's okay. We'll talk about, we'll touch on this. So this is in the center here, and this feeds off it precursors, intermediates all over the place. There are also other pathways here that are related to each other. So everything's connected to everything else. And as a microbial physiologist, if you know the beginning of a pathway, you can usually figure out how the end of the pathway is gotten to on the basis of the structure and the kinds of reactions that occur. Anyway, we're not going to be focused on that because I can't do this in 2 lectures. It would have to be basically a whole year's worth of a course to do this. But I just want to get you a feel for this. I used to joke that you guys would know all this by the end of the semester, but that's not. You know that isn't going to be true. Okay. Oh, I know what I wanted to do. Let's go back a second. So I want to, and I apologize for doing this, but I have an empty slide here, and I wanted to show you this slide for 2 reasons. So let me share my screen and show this slide. Okay, let me know if everybody can see this. There should be 3 panels. Okay, great. Okay, so the panel on the left here, let me get that started. What's happening? So if you don't know, and you were confused about this from the last lecture, this is called motility. Yep. So these bacteria are swimming, and it looks like they're randomly swimming all over the place. If you just focus on one, and I'm not sure you can see a pointer here, focus on one, it's just basically going in one direction, and once in a while it'll turn around and go in the other direction. So this is what the flagella are using, are help having, and actually they're bumping into each other and they're changing directions. Okay, so that's one thing, that's motility. This is a culture of bacteria that is doing the swarming phenomenon. It looks kind of eerie, but this is a whole culture bacteria moving in one direction towards something. So these cells are swarming. And finally, and I'm not, for some reason I can't get these to go all at the same time. So and I'll do this last one here. What are these guys doing? Right, so they're dividing. Binary fission, absolutely right. Okay, so what do all of these three things have in common? I basically gave the answer away, but yeah, they use energy. Well, so dividing doesn't require movement. Dividing is basically something that occurs from the inside. So the two top ones are movement, but the bottom one is not movement. It's just division, and the cells look like they're moving. It's only because they're taking up space as they're dividing. So all three require energy, and in order to get this energy, basically the idea for this division here is that's the essence of being for bacteria. That is what is responsible for making bacteria viable, meaning that they're able to grow and divide. An organism that is not capable of division is not viable. And in order to do this, it has to metabolize. It has to bring in a source of carbon and a source of energy, and a variety of other things. Okay. All right. Any questions about that? I'll go back to our slides. Questions? Am I back to you guys on your desktops here in terms of components of metabolism? Or am I still on my desktop? We're back to class, right? We're back to my lectures. Okay, good. For some reason I'm seeing everything on my desktop as well, but it's okay, no problem. Okay, so components of metabolism. I want to spend the remainder of today talking about enzymes. We're going to talk about ATP, electron carriers, chemical energy sources, precursor metabolites in the next couple of lectures, and their relationship to each other. I don't want to spend any time talking or introducing them right now because we'll do that as we're going along. So let's talk about enzymes. Okay, so you should all know that these are biological catalysts. What does that mean? What does it mean that they're a catalyst? Okay, so they help speed up reactions. That's correct. But what the major part of that is that they don't participate in the reaction itself. That is, they don't participate in the reaction in the reactants or the products, right? They're basically silent, and they facilitate each step of the metabolic pathway. So you have an example of metabolic pathway here, and I don't know why that came out so crappy like that. So each enzyme, each reaction in the pathway utilizes a different enzyme. And yes, they decrease the activation energy. So without an enzyme, it takes a certain amount of energy to go from the reactants to the products. And with the enzyme, it decreases this. And I know those of you who have biochemistry, this is very basic material. For an enzyme reaction, instead of talking about reactants, we talk about substrates. These are the compounds that enzymes bind to catalyze the reaction. So it accelerates the conversion of substrate. So starting compound here, enzyme A takes that and makes this product, intermediate A. Enzyme B takes this intermediate A and makes intermediate B, etc. The place where the substrate binds to the enzyme is called the active site. And the enzymes, because they're made up of amino acids, they're not fixed in space. They're able to actually change shape. So the binding of a substrate to an enzyme involves a shape change of the enzyme, and that shape change actually, during the course of this binding, it's called an enzyme substrate complex. That shape change affects the release of the products. So if you have two substrates that are in a reaction, so substrate A and B come together in the active site, they'll come together and form the product, and the product will be released. And that's how it occurs during a shape change. And a lot of enzymes have these cleft-like structures where the active site amino acids are involved in binding of the substrate and catalyzing the reaction. And you can see what happens here is this enzyme is now more compact as it bounds substrate. The shape change is referred to as conformation, or the ability to change shape. And so the enzyme is in one conformation in the absence of substrate, and a second conformation when the substrate is bound, and then once the substrates are left, it goes back to the first conformation. The active site, as well as all these, so these balls and these balls that you see here are all different colors. These are all amino acids that are held together in the protein or the enzyme by a variety of different forces. Anybody remember the kinds of forces, or give me one kind of force that's used to hold together this enzyme in this way? Ionic bonds, correct. That's one way of holding this that's held together in a certain conformation. Any others? Hydrogen bonds, that's correct. And there's a couple more. Disulfide bonds, disulfides are usually what's held together is intramolecular and then different subunits of an enzyme together. But yeah, that does hold it together in a certain way. Covalent is basically the backbone of the amino acids. So that's not something that's fixed. When this thing is put together, the covalent structure is basically maintained. It's how the amino acids interact with each other. So yes, hydrophobic interactions, that's correct, and hydrophilic interactions are the other ones. So we could do this poll here. And give me a minute to set it up. Which of the following factors do not affect enzyme activity? Another minute. Okay, so temperature, salt, pH are all factors that affect enzyme activity. Metals affect enzyme activity, and in fact, we'll talk about how metals affect enzyme activity in a couple minutes. Humidity does not affect enzyme activity. Enzymes are generally associated with aqueous environments, and they require a certain amount of water, and humidity is associated with air. So that would be the wrong answer. Okay, so here's the factors that influence enzyme activity. Basically, enzymes have narrow ranges of biological activity, so the ranges in certain temperatures. So here between 35 and 45 is the optimal temperature for this group of enzymes. Enzymes that are optimum pH here. Now, you have to recognize, the answer for the last question was humidity was the one. You have to recognize that conditions here, optimum temperature, optimum pH, are not all enzymes. An organism that grows at a temperature of 100 degrees, the optimum temperature for the enzymes in that organism would generally be around 100 degrees. Similarly, an organism that grows down at pH of 2 to 4, the optimum pH conditions for that enzyme would have a shape that would look like this. So actually, let me use a marker here. So it would have a shape that looks like this, rather than this. And an organism that grows down at 20 degrees would have optimal activities somewhere in this range. So just recognize that temperature influences activity depending on where the organism is from and the enzyme that you're talking about. And the other thing that affects enzyme activity is substrate concentration. That is the substrate concentration. As the substrate concentration increases, and that's the reactant increases, enzyme activity will go up until it gets to a certain point. And then no matter how much substrate is there, you're going to have, excuse me, you're going to have a point where no substrate is going to change the activity. It's going to be at maximal saturation, maximal enzyme activity. Metal was not the answer to the last one. And if I can go back, I said humidity is the answer to this. Humidity does not affect enzyme activity. Metals do. Are people getting kicked out? Is that what's going on here? I thought what I was getting was Okay, well, this is a Blackboard issue. I'm going to have to. Does this help? Those of you who are doing Collaborate in other classes, do you have this problem? I wonder if this is the carrier that you're using might have something to do with it. It might be those of you who are having this problem. Yeah, it could be the Wi-Fi of where you're at. I'm certainly not getting kicked, but that doesn't mean anything, right? I'm sorry if that is occurred, but that's the usefulness of having the recordings. And if you miss something and you come back in and you want me to clarify, I'm happy to clarify it anyway. I'm sorry. Again, the technology should be really great, right? But it depends on a lot of other things. Okay, so let me go back here. So what happens? What happens at this point here? You get to too high a temperature. When you get, too high pH, or you get to too high a temperature. And actually down here when you get too low pH. And the answer is basically because an enzyme has a particular shape, a particular conformation, going above and below the pH that it functions at optimally, or above the temperature that it functions at optimally, you end up getting a disorientation or disorganization of the backbone chain, the amino acid chain associated with it, and you get the denatured, becomes denatured. Sometimes a protein can denature. You can go this way, that way, it's reversible sometimes. Most of the time when it goes this way, it can't go this way. That's a classic example of this, and one that you all know is when you boil an egg, the egg white of the egg becomes from a clear to white, and the white is because the proteins have gone from the functional form, denatured form. The denatured form comes out of solution, basically comes dissolved out of solution, and it can't go back to the functional form. So why you can't make an egg white go clear. Okay, questions? Stop there for questions. All right, let's go on, talk about the biological control of enzyme activity. There's 3 different ways. book has talks a little bit about something called allosteric enzymes, but I like to feed them. I like to stick them into here, as you'll see. The basic way of controlling enzymes activity. What we're talking about here. Let me put this in context. We're talking about a way that you have the substrate to one intermediate, to a third intermediate, to a fourth one, then you get a final product, and that product then is used for a synthesis or something like that, protein synthesis. Okay. Each step has an enzyme. This is enzyme 1, enzyme 2, etc. That catalyzes the reaction. So if there was no control of any enzyme in this process here, the cell wouldn't have a way to speed this up, slow it down. Say, let's say that it's got enough F that it needs to make DNA. How would it actually turn off this whole thing to slow it down so that it doesn't waste any more of that A on the way to F? Alternatively, if it doesn't have enough F, does it speed up going from A to F? So you need to control the enzymes, and these steps here are different ways of controlling it. So competitive inhibition. This is a very simple one, and it is basically self-explanatory. That is that the inhibitor competes the active site. It binds to the active site of the enzyme. The inhibitor binds to the active site of the enzyme. The structure is similar to the substrate. And this means that the more you add, more substrate will block, so the more the enzyme's activity will decrease. And so from the pictures here, what it's using is a classic example of an enzyme whose activity is needed for making a group of compounds called folic acids. I'll write that down. Folic acids are vitamins, are cofactors that are involved. And I just realized when we go back to the structure of the enzyme, I didn't talk about this, so I'll necessarily have to say something about this in a minute. These are structures that are needed for certain reactions. And to synthesize them, you would need this one enzyme that uses this compound called PABA. And it's not really important to know what PABA stands for, other than I'm going to write it, but it's not. Benzoic acid. So Stefano asked the question: how long does it take for an inhibitor to debind from an active site? So to be released from the active site, and actually for this process here, binding of the inhibitor to the active site is very similar to the binding of the substrate to the active site. When the substrate binds, the substrate can come off the active site just as easily as the inhibitor can come off the active site. Inhibitor, since it doesn't go to product, actually be released from the enzyme because it can't be made out product. So it doesn't depend on the substrate concentration per se. It depends on the binding constants, the compounds. And I don't want to get into that into a lot of detail because this is not the course we're talking about that. But each inhibitor and each substrate has its own binding constants for the active site. And competitive inhibitors that are really good have very similar binding abilities to the active site, just like the substrate does. So they basically compete with each other for that site. In this case here, what I wanted to talk about was the fact that there's a drug called sulfanilamide. It actually looks like para-aminobenzoic acid. So this is what binds to the enzyme as a substrate. And this inhibitor here, the differences between the two, everything else is the same. But the difference here doesn't allow this to be used as a substrate. The enzyme will take this and use this as the substrate, but this will inhibit the activity. It binds to the active site just like the PABA does. So this is a classical competitive inhibitor. This this is a drug that you find in drugs called sulfa drugs. Sulfanilamide is one. Bactrim. If you've ever had an infection that required the use of Bactrim, Bactrim contains this kind of drug, these sulfa drugs. They're used for urinary tract infections. They're used sometimes they're used for lung infections. Depends on the infection. The question I would have to you is, why would sulfanilamide be potent against these kinds of enzymes? Why wouldn't they affect us? We have folic acids in our bodies. We use folic acid. We require it. If you look at the side of a cereal box, you'll actually see at times there's folic acid in them. The question is, why do sulfanilamide and the sulfa drugs not affect us? Any guesses? Well, folic acid has nothing to do with the cell wall, Jonathan. It's this has to do with compounds that are involved in biosynthetic pathways, generally involved in making pyrimidines. So folic acid is a coenzyme. That's correct, but that's because, and that we need it. So the question is, why is its synthesis, when you inhibit its synthesis, why doesn't that occur? Why doesn't that drug work in us? Somebody made a comment about cells transport them in the cell a different way, and that could be one way that the drug might not affect us because it can't get into the cells, but that doesn't happen. Coenzymes and cofactors are identical, so these are cofactors or coenzymes. We need them for some enzymes to work. So Anna came up with the answer is, we don't make our own folic acid. Folic acid is provided to us in our diet, the bacteria in our guts, or by foods that contain folic acid. And that's why we don't have to make it, and why sulfanilamide doesn't affect us. And I'm gonna have to go back here for a minute, and actually I'm not gonna go back. Let me go back. Excuse me for a second, and I apologize to do this. I'm going to go back to a slide because I neglected to talk about, where are we? Cofactors and coenzymes. So yeah, that's what's missing. And let me see if I can do this. I'm going to go all the way back to the structure of the enzyme, and I neglected to talk about this. Yeah. All right. So here's where I went ahead too fast. I want to talk about this guy here, cofactor. So substrate binds to the active site. The enzyme has amino acids in here that interact with the substrate. But there are some enzymes that require some other factors that interact with near the active site that are used to bind the substrate. These are called cofactors or coenzymes. And I'm going to have a tough time writing this way. And their other names for them are often vitamins. Not all vitamins are cofactors or coenzymes. Some of them are. But, these are required by some enzymes to help assist the binding of substrate and catalyze the reaction. Folic acid is an example. Is not an example of one of these. I'm sorry. Folic acid is an example of one of these, and there are a lot of others that are associated with the binding of enzymes with substrates. And all I wanted to say was that certain cofactors that are also involved in metabolic pathways, and we're going to be talking about the reduced and oxidized form of the nicotinamide, NAD. And the flavin adenine dinucleotides, FAD, and coenzyme. All right, so AD, NADH is the other one, ADP. These are cofactors or coenzymes bind to the enzyme's active site, near the enzyme active site, and help in the reaction that the enzyme catalyzes. Okay. Let's get back to Anna. Did you have a question? All right. Okay. All right. That's all right. No problem. So, last couple of minutes here. I just want to finish up on enzymes. We talked about competitive inhibition. Basically, the inhibitor looks like the substrate and binds to the enzyme active site. Non-competitive inhibition, the enzyme has a separate site. That site is called an allosteric site, where the inhibitor or the compound, the controller, changes the shape of the enzyme so that it now no longer is able to bind substrate at the active site effectively. There's two ways of doing that: a negative inhibitor or a negative allosteric inhibitor. Sorry, an allosteric inhibitor that negatively affects the enzyme, so it binds, distorts the shape, and now the substrate can't bind. So the site other than the active site is called the allosteric site. This is where the allosteric inhibitor binds. There's also allosteric activation, where a compound will change the shape of the enzyme, the conformation of the enzyme, that it is now able to bind substrate better than it did before. So this is called activation. Both of these cases, the allosteric enzyme, that is the enzyme that has this extra site that's affected by these molecules, is at the beginning of a metabolic pathway. So if you have this long metabolic pathway, in general, the first enzyme of the pathway, and so I'm going to just draw that real quickly. So here's the pathway. Here's the first enzyme of the pathway. This enzyme here is generally the enzyme that's going to be either stimulated or inhibited by the binding of an allosteric compound, the allosteric site. And the reason for that is that sometimes the organism makes an enzyme that doesn't work very well, and it needs to have a stimulator to make it go faster. Sometimes it makes and so it changes its conformation. Sometimes the enzyme is made in a really active form, and the organism wants to turn it down very quickly, changing the active site. So that's the non-competitive control. And again, the non-competitive is exactly stated that way because you don't compete at the active site. You actually bind to a site other than the active site, referred to as the allosteric site. And finally, feedback inhibition, where enzyme activity is controlled by binding at the active allosteric site, just like what we talked about. The regulatory molecule that occurs is an end product pathway. So you have a long metabolic pathway. The end product comes back, feedback inhibits binding to the activity by the allosteric site of the enzyme by affecting the enzyme so that it now turns down the activity of this pathway. Sort of like turning off a nozzle spigot to make the water flow less, or turning it up to make the water flow faster. Any questions about this? Right. So if you have questions, I'm in my office hour tomorrow, and I'm certainly available by email on Blackboard. And what we'll do is we'll finish here now, and we'll continue taking up ATP and central metabolic routes on Tuesday. Have a great weekend. Enjoy the Super Bowl. Enjoy Valentine's Day, and I'll see you all on Tuesday.